So again, a warm welcome, everybody, to this seminar, the fourth one, I think, in this series, where I am going to give a little bit of an overview and provide you with some details on some of the projects that we have ongoing and are working actively with in the lab. On the computational side, to try to understand how, essentially how we can use computational tools to understand the performance of lipid-based formulations and the interplay between drugs, formulations, and the different kinds of rearrangements that goes on in the human intestine. And to do so, we use specifically a computational tool called molecular dynamics simulations. And I will just briefly mention what it is and how it works. And just to give you a flavor to start with, this is a quote, quite a famous quote by Richard Feynman, who was a American theoretical physicist. And then, so he said that all things are made up of atoms, and everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And so if one is to essentially conceptualize what molecular dynamics simulations is, one can say that it is a technique essentially to in the computer study the jiggling and the wiggling of atoms over time. And obviously also the 2013 Nobel Prize in Chemistry was awarded to Martin Karplus, Michael Levitt, and Ari Warshel for the development of multi-scale models for complex chemical systems, which at the heart of it is essentially MD simulations. And just some more foods for thought, why we want to do simulations, why they can be relevant in studying a particular problem. And so I've listed here a number of arguments, I guess one can say, for why we want to do simulations. And so in some cases, the experiment is simply impossible to perform. For example, you can't simply go and look inside of a star, or you can't perform a sensible experiment with the expected outcome of forecasting the weather. It can also be too dangerous. Flight simulations and different kinds of explosion simulations and nuclear simulations of nuclear reactor processes fall into this category, I think. Some experiments are also prohibitively expensive. High pressure simulations, wind tunnel simulations. And perhaps most relevant in this context is that some experiments are simply blind because the properties of a particular system that we are interested in cannot be observed on very short time scales or very small length scales, even though they occur on those time and length scales. But we can't see them experimentally, or at least we can't see them without a certain degree of difficulty. So simulations then is a very useful component in any scientific endeavor, really, because I think it can replace experiment, it can provoke experiment, but it can also, in some cases, explain experiments. So it's a little bit difficult to introduce MD simulations without going through, or one can introduce it without and introducing lots of equations and so on. And I'm not going to do that here in the interest of time, but instead just show you a small movie of what it looks like when one is simulating. In this case, this is an MD simulation of a number of water molecules that are interacting over a certain period of time. So you can see the red spheres, that's the oxygen atom, obviously, of the water molecules, and then we have the hydrogens, the white spheres, and then these yellow dashed lines are the hydrogen bonds between the different water molecules that are being formed and broken up over the course of the simulation. So this is a rather short simulation, and it's perhaps a bit uninteresting in the sense that it's only water, but it illustrates the general concept of what the simulation looks like, or what you essentially get out of the simulation. So again, as the quote was also made by Feynman, the jiggling and the wiggling of atoms over time. but it's not just for water, as I have tried to say. It's being used in many different scientific fields, perhaps most prominently in the study of protein solvation, or at least protein folding, but also for things like DNA strand dynamics, certainly within the context of membrane dynamics, drug discovery and drug delivery. It has been used for the study of crystalline and amorphous solids, different kinds of dynamic processes involving various metals and organic frameworks. And also a lot of research has been done in terms of trying to understand the dynamics, what goes on at different interfaces between different materials and so on. So as long as you can formulate any kind of description about the system that you're interested in on a level of individual atoms, essentially, in theory at least, you can use MD to study that process. But so what I will do now, so that was a little bit of introduction and setting the scene, talking in general terms about simulations. So what I will do now in the next half hour or so is to just show you examples of 3 different avenues of research that we have performed at the Department of Pharmacy, where we have specifically used molecular dynamic simulations to try to understand different aspects of these systems, essentially. So the first thing I want to cover is some work that we have been doing on simulations of lipid-based formulations. And then the second part of my talk will be about the influence that lipids and bile salts have on drug partitioning, specifically into lipid bilayers in this case. And then finally, towards the end, or the last third of my talk, is going to be on studies on simulated intestinal fluids. And obviously all of these things are connected, because what happens when you administer a drug that is intended for oral administration, it might be formulated in a lipid-based formulation, it's ingested, digested in the small intestine, which does contain a lot of lipids, a lot of bile salts, and those components form various kinds of colloidal structures that in different ways contribute to the final behavior of the drug, and essentially the bioavailability of that particular drug. So the first example I wanted to tell you about are the simulations that we've done on lipid-based formulations. So here we have used, as it says, MD simulations to investigate the nanoscale behavior of lipid-based formulations with and without a few different drugs. And some of the specific questions that we have tried to answer, you have them listed there as well. Are there any structures formed in these formulations? If so, do the drugs interact with these structures? And can we use this knowledge in some way to say something intelligent about how these lipid-based formulations will perform in terms of drug solubilization? And with structures here, I mean any type of sort of aggregation of lipids in general, micelles, inverse micelles, lamellar phases, hexagonal phases, so any shape that, or any aggregation state essentially that the lipids can display. So this is the setup that we had in this particular study. So we have studied 4 different lipid-based formulations. You have them here on the top of this slide. So they are named, they are categorized into different types, depending on essentially the amount of lipids that they contain. So there's also, I should also add that there's, so here we have type 2, type 3A, type 3B, and type 4. There's also a type 1 lipid-based formulation which contains only lipids. But in this case, we are focused on type 2, 3, and 4. So as you can see here, type 2 then contains, for the most parts, lipids, going over to type 3A and type 3B, in which the lipid content is reduced somewhat, and you add in other additional surfactants and excipients. In this case, carbitol and PEG 400, or carbitol and polysorbate 80 in the case of the type 3 lipid-based formulation. And then finally, we have the type 4 lipid-based formulation, which essentially does not contain any lipids at all, at least in this case, but rather is made up of only surfactants and other excipients. So we have simulated each of these formulation types at different amounts of water. I think there's also a 75% water simulation for these. And we have simulated each of these cases with 3 different drugs, tolfenamic acid, progesterone, and noscapine, selected because they have different partition coefficients, and also that they are different chemically, in the sense that one of them, tolfenamic acid, is obviously an acid, and the other ones is a basic drug and a neutral compound. So, as I said, one of the things that we tried to investigate is to see if there is any structures of some kind formed in this lipid-based formulations, and we initially focused on the dry lipid formulations, because it has been described in the literature that solubility, the final solubility for a particular drug in a lipid-based formulation, essentially is to a good approximation at least, is a linear combination of the solubility of that particular drug in the individual components of the that make up the final formulation, weighted by the concentration of the individual formulations. And so we then had the hypothesis that suggests that even in the dry formulation, so before adding any water, that there should be nanoscale structures with which the drug molecules can interact, and that would help to explain why we see this linear combination behavior in terms of concentration or solubility, the connection between concentration of the individual excipients and the solubility. So this is a type 2 lipid-based formulation. Again, the majority of the components is lipids, and then we have another 20%, which is, in this case, chemophore, which is this molecule over here. So it's a polyethoxylated oil molecule, essentially. And what you see here basically are snapshots of the simulation after a certain amount of time, I think a few hundred nanoseconds. So these are identical images, except in the right one we have removed the chemophore for clarity. And what you can see, or what I would like to point you to, are, for example, regions like this, where we have a space here essentially that seems to be devoid of. So as you can see, the lipids here are colored in teal or in teal, or in light orange, with the head groups, the oxygen and one hydrogen as a red and white sphere. But in this region, for example, we have a region where we have no head groups. So this is essentially some kind of, or this indicates at least that there is some kind of inverse micelle being formed here transiently. It overlaps to a certain extent with chemophore. So the chemophore molecule seems to be filling this space to a degree, but not completely if you analyze it in detail. And if you look a bit more carefully, you can find a number of structures that look like this. So essentially microscopic structures that exists even in dry lipid-based formulations. And so in this case, there's also a single water molecule present even in the dry state. So some of these lipid molecules contain, or lipid-based formulations contain trace amounts of water, even when they are considered to be dry. So we do see formation of what looks like, at least visually, to be small inverse micelles formed by the lipids in these simulations. And when we add water, so this is the same type 2 formulation, adding in 5% by volume or by weight of water, and then adding in 30%, we obviously drive the system more and more towards a a phase separation. In the case of 5% of water, there is the components still appear to be reasonably well mixed in the sense that we don't see any single clustering of the water molecules, at least on the time scale that we have simulated. Whereas if we go up to 30% water, we start to see a phase separation. And it's a little bit tricky to see here, but if you look at this system in detail, you see the chromophore molecules sort of orienting themselves in the interface between the water molecules and the more hydrophobic lipidic phase that is formed away from the waters. And again, we see small inverse micelles formed by the lipids in these simulations as well, that contain, at least at these small amounts of water, small pools of water being formed. So this behavior is maintained even when we start to dilute the systems with water. And then obviously we also added in, as I said, we added in drugs. So I will show you one example of that here, again, for the same type 2 lipid-based formulation, where we have again removed chromophore for visual clarity. And this is when we have added 20 molecules of tolfenamic acid to the formulation. So as you can see, this is the final frame of the simulation, where we have the drug molecules sort of evenly dispersed in the simulation box. And so again, we set out to look for if the drug molecules interact with any kind of structure in the simulations. And so this is out of the 20 tolfenamic acid molecules that we have inserted in the system, I pulled out 15 of them. And the other 5 look the same. So as you can see, in all cases, the drug molecule surrounds itself again with what is resembling some kind of inverse micelle type structure. So it would seem that the drug molecules do indeed interact with well-defined local regions in the formulations. I should also add that these assemblies appear to be somewhat transient, so they come and go over time. So these are just snapshots. But at least in some of the simulations, we see these inverse micelle type structures and the drugs interacting together. We also see that we get different behaviors for different drugs within the same lipid-based formulation. So this is another example, or this is an example of that. In this case, it's not tolfenamic acid anymore, but instead noscapine. And instead of a type 2 lipid-based formulation, this is now a type 3A, which contains 50% lipids and then 30% polysorbate 80 and around 20% or so carbitol. And so as you can see here, so this is an average over all 20 drug molecules. So again, we have 20 drug molecules, noscapine instead of tolfenamic acid. And then we have overlaid each of those 20 molecules, and then we have quantified the local environment in terms of the spatial density and temporal density of where molecules in the lipid-based formulation, so where the lipids, the polysorbate 80 and the carbitol like to sit in terms of interacting with the drug molecule. So as you can see here, at least for noscapine, we have one side of the noscapine, and then we have the other side of the drug molecule, which interacts similarly with the polysorbate 80 and the carbitol. So it seems like, to conclude this first part of the talk, molecular simulation can be used to show formation of local order and microstructure in the lipid-based formulations. It seems like the drugs in the formulations interact with these structures in different ways. We need to explore this a bit more. And we also need to think about I think, essentially concrete measures, numbers that we can put on this instead of, as I have done now, showing images of how it looks like in the simulation. And the question still stands if we can use all of this to make any quantitative predictions about what the solubility of a particular drug will be in a particular lipid-based formulation. So with that, going further and relating to the second part of my talk, we were also interested to see what happens to these systems when we introduce components of human intestinal fluids. So we have a study where we introduced two such components, one so taurocolate, which is bile salt, and also lipids in the form of DLPC lipids, so a phospholipid. So these are those two components again. So taurocolate on the right-hand side, and the phospholipid on the left-hand side, which has a couple of double bonds on the aliphatic carbon chains. And so we did a study here where we tried to assess the influence, the degree to which partitioning of taurocolate molecules into or affects or is inserted into lipid bilayers. So what you see on the left here is essentially on the right hand side. So this is the free energy. So the amount of energy that is required to insert taurocolate molecules into a lipid bilayer that is made up of this other phospholipid, and where that bilayer also contains either one or two or three or four taurocolate molecules previously locally around where we insert the additional taurocolate molecule. So as you can see, initially the red line, the green line here, it's the delta G is positive towards the center of the bilayer with about 10 kilojoules per mole or so. So that indicates that initially it's unfavorable for bile salt to partition into lipid bilayers. But then there seems to be some kind of cooperative effect taking place. So upon insertion of additional taurocolate molecules, you get a significantly reduced free energy in terms of inserting other taurocolate molecules into the lipid bilayer, until you reach a point where you have about 3 to 4 taurocolate molecules already inserted into the bilayer, at which point you don't seem to be gaining any more free energy. And we also investigated in this study the influence of taurocolate molecules not inserted into the bilayer, but so floating around in the water phase of the lipid bilayers. And then we could see that also has an influence on the insertion free energy of taurocolate molecules. So if you compare, so the red line and the purple line, so XTCH stands for extra taurocolate, extra as in outside of the water phase. So it makes a little bit of a difference to have taurocolate molecules also floating around outside of the lipid bilayers themselves. And this is one thing that we see. So this sort of highlights what I said about the fact that we see a stabilization in the insertion free energy going from about 3 to 4 inserted taurocolate molecules. So when we have 4 taurocolate molecules inserted close by to one another, it looks something like this in a model lipid bilayer. It's quite clear when we analyze the hydrogen bonding patterns between those taurocolate molecules that these clusters that then span the entire bilayer are stabilized by formation of intramolecular hydrogen bonds. And that would also explain why we don't get an additional effect by adding in more taurocolate molecules, because there's no more hydrogen bond donors or acceptors available for these taurocolate molecules to stabilize further. So this is relevant, of course, because it is through these bilayers. This is a model of the bilayers, but it's through these bilayers that the drugs that we are administering orally need to pass in order to reach systemic circulation. So then we, in the final part of this study, we took 3 model compound drugs, carbamazepine, felodipine, and danazol. And we investigated, again, the free energy of insertion for these drugs. We also had, in this study, included water and ethanol for comparison. And we compare the situation where we have no bile salts in the membranes with the situation where you have bile salts configured as on the previous slide in these clusters of 4 by 4 molecules of taurocolate. And so as you can see here, there is a difference in the insertion free energy of the drug molecules with and without taurocolate. So for example, for danazol, it seems to be preferring to insert itself into bilayers that contain taurocolate. The minima of this PMF is also shifted a bit towards the center of the bilayer, so that indicates that the danazol molecules will also partition a bit deeper into the bilayer. And we also get quite a good, or a decent at least, agreement when we calculated the partition coefficients for these systems. So it seems that the amount of bile salt, we do know that the amount of bile salt in the human intestine varies, and this has consequences for drug absorption. And the way that taurocolate, in our case, interacts with lipids may explain part of the observed inter-individual variation in drug absorption, in the sense that we get different absorption profiles, depending on both for taurocolate itself, depending on how much taurocolate we have free in solution, but also for the drugs, depending on how much taurocolate we have previously inserted into the membranes. And this affects different drugs differently. So this leads up to the final part of my talk, which is about studies of how this connects, or where we have tried to perform studies to connect this to the intestinal fluid itself in greater detail. And so there's been a number of, as I'm sure you're aware of, a number of simulated intestinal fluid compositions developed with the intention, or with the aim, of mimicking fasted and fed state intestinal conditions to allow for simpler determination of in vitro drug solubility. So we set out to do this in a collaborative study involving both MD simulations, but also small angle X-ray scattering, dynamic light scattering, and cryo-TEM, where we have a number of different simulated intestinal fluids that were characterized in terms of colloidal structures and the sizes and shapes of those structures. So in this study, we included systems that contain only bile salts, mixed micelle solutions, and then commercially available powders with simulating or intended to replicate, again, the fasted and fed state intestinal fluids. So these contain taurocolate again and lecithin. And these ones were made in-house, containing, obviously in the case of bile salts, only taurocolate, and in the case of the mixed micelles, taurocolate and not lecithin, but another phospholipid, DOPC. So I won't go into the SACS results that much, because this is intended to be about focusing on the computational parts. But what was clear, one of the outcomes from measuring the colloidal structures, or analyzing the colloidal structures with small angle X-ray scattering, was that a lot of those colloidal structures were found to be ellipsoidal in their shape. And the mixed micelles, so the micelles containing both lipids and bile salts, were found to be larger in general than the bile salt only micelles. And the best fit in terms of modeling the SIFS data was when you used a model that had a core shell type structure. So where we had a core of lipids and then a bile salt shell on top of that. And the general size of these micelles were on the order of about 5 nanometers or so. So what we did in the computer for this study was simulations of these 6 different simulated intestinal fluids using the GROMACS software and the Martini force field. And so this is always, when you do computational simulations, there is a trade-off that you have to make, because you cannot, because these calculations tend to be quite compute intense. Typically, you cannot simulate precisely the type of system that you want at the exact detail that you would also want. So in order to simulate actual physiological concentrations of these components, we had to make use of so-called coarse-grained molecular dynamics simulations, which essentially means that instead of having the atoms explicitly represented, having all atoms explicitly represented in the system, we instead lump a certain number of heavy atoms together into larger coarse-grained beads. So this is an example of what a lipid molecule, in this case DOPC, looks like in the Martini force field. So instead of having a long carbon chain with 16 individual carbon atoms, or 18, we now have instead 4 coarse-grained beads. So obviously then the effect that this has is that we need to calculate fewer interactions per time step in the simulations, which means that the simulations progress faster, essentially. So these are some of the results. For the fasted and fed bile salt micelle systems, and then the mixed micelle systems, and finally also for the FESIF system. So as you can see, in general, the sizes agree fairly well with what one would expect from this small angle X-ray scattering measurements. The bile salt micelles A and B, you have a scale bar here, so this is 2 nanometers. So they are elongated in 1 dimension, and the sizes, so they're a little bit on the small side, perhaps compared to the experimental data, but in general, the agreement is quite okay. And we also see that we have the mixed micelles C and D appear to be larger in general than the bile salt micelles. So that's quite good. And again, just to get that point across, so this is a comparison between the scattering data. So this is the normalized scattering data expressed as the interparticle density, the probability density. So where you have a peak in this plot is where you have the majority of sizes coming from the colloidal structures of the scattering experiment. So the radius appears to be around 4 nanometers. So again, we are a little bit, in comparing the same system in the simulations, we're a little bit on the small side, but I think in general the agreement is quite decent. And again, to show all of the systems in the same slide, we have also calculated shape factors. So the idea is to be able to differentiate between spherical systems and things that are more ellipsoidal. So a sphere would have a shape factor of 1. And anything that is deviating from a sphere will have a shape factor greater than 1. So these are the shape factors coming from the SIFS experiment. So about 1.8 or 2 for the fed and fasted bile salt micelles, which appear to be slightly more elongated than the mixed micelles. and then SIFS at 1.5. So comparing that, so these are the simulation data. So the fed and the fasted bile salt micelles, we have some structures that do seem to be more elongated than average. And then for SIFS, again, the shape factors appear to be somewhere between 1 and 1.5. So qualitatively, the agreement is there, and the same is also true for the sizes, which I don't think I need to talk about this. I've covered that before. But so we see that the largest structures expressed now in volume instead come from the fed and fasted micelles, and this is also something that we see in the simulations. So the question is then, what do all of this, what consequences does all of this have for any drug that you administer into these intestinal fluids? So to do that, we took the experimentally determined intestinal fluid composition from 6 healthy volunteers. So this was experimentally determined compositions. We first simulated the actual concentrations again, and then we extracted individual micelles from all of those 5 simulation series. And we performed new simulations, adding in 2 drugs, prednisolone and fenofibrate, at different concentrations, so 6 different concentrations per drug. And if you do that, and if we also normalize for the concentration, the difference in concentration for these different drugs, and we have calculated here the number of contacts between the drug molecules and the micelles. So we see that for irrespective of the intestinal composition, or the composition of the intestinal fluid, we see that fenofibrate for all cases seem to have a higher affinity or a greater affinity for the micelles than prednisolone does seem to have. So that would suggest that the solubilization enhancement for fenofibrate, or fenofibrate is benefiting the most from having formation of various kinds of colloidal structures over prednisolone. So to conclude here, it seems that coarse-grained simulations of human intestinal fluids resulted in self-assembly of bile components into ellipsoidal micelles of reasonable size for the smaller colloidal structures. I should say I didn't present data for the case of SIFS, which from the SACS experiment formed much larger structures on the order of several tens of nanometers, and up to, I think in some cases, 100 nanometers. And obviously, even with coarse-grained simulations, we cannot get to that size yet, at least. And simulations of these micelles, that was the result of this set of simulations, in combination with fenofibrate and prednisolone, resulted in fenofibrate having more interactions with the micelle molecules compared to prednisolone. So one would predict, based on that, a higher effect, or a larger effect on the solubility for fenofibrate than for prednisolone, given what we can say about the composition of the intestinal fluid. Right, so just finally, where can we go with this? So I think we're now at a point where, or as I've tried to show you, simulations provide, or MD simulations provide insights into time and length scales that are not otherwise easily accessible. And it can be used, or they can be used for coming up with molecular level explanations of drug solubility and partitioning. And also we have started to explore a little bit around the impact of the intestinal, of the inter-individual variability of the intestinal composition in terms of how that affects drug solubility and drug partitioning. And I think finally, as we have realized over a number of years now doing this, computers are finally becoming powerful enough to simulate sort of pharmaceutically relevant systems at actual concentrations. Obviously, you can simulate smaller systems, but if you want to make the argument that we are simulating at actual physiological concentrations, I think we're finally now at a point where we can do that, essentially. And so with that, I just want to end by acknowledging the people who have done this particular work: Albin Perro, Linda Alskär, Shakawat Hussein, Alexei Kaberev for organizing this series, Christel Bergström and everyone else in the drug delivery group at Uppsala University, and people at Umeå, Lancaster, and the Boyd Lab in Monash for a good collaboration on the SAX study. So with that, thank you very much. I think there should now be time for some questions, right?